We'll continue with NGS in proteomics, which stands for next generation sequencing in proteomics. Why do we do this and what is this? So I never thought when I was in university I would actually do the same as my teachers and start with this, which is basically the central dogma of molecular biology, that you have DNA, RNA, and a protein, but it actually makes a nice entry into a presentation. So I'm also going to do that. So as you all know, we have DNA that stores the information about everything in our cells, and then DNA gets transcribed into RNA and then translated into a protein. And we have different techniques to work on the different stages of this process. So on the DNA level, there are also different things we might be interested in than on the protein level. And we can study different aspects. So this pyramid shows kind of what we can study or what we might be interested in on these different levels. So on the DNA level, we might have copy number variations and mutations, methylations, and so on, which might or might not affect the protein expression in the end, but also might, for example, affect the translation process. So maybe the DNA is not like the transcription process is not exactly the same as then resulting in final proteins. On the RNA level, there's also happening a lot like splicing, modifications, the expression itself. We could, in theory, also study the secondary structure of the RNAs and so on. And then, I mean, what most of us do here is studying proteins, so proteomics. So you can study all these kinds of things and much more that you do in your fancy research. So next generation sequencing basically is just a high throughput sequencing of DNA. If you reverse transcribe RNA into DNA, you can also do this with RNA. So basically NGS gives you information about DNA or RNA. So this is what this talk is going to be about. So how do you combine this with proteomics? And maybe we should do this more or maybe not. So this is actually a very nice picture that a PhD student in our group did. Now he left, but it shows this process on different scales, and I really like that. So we have at the like lowest scale, or however you might call that level, you can study the chromatin when the DNA is still packed here, and you can study all kinds of DNA binding proteins, the chromatin structure, epigenetic markers, and so on. And then, so this thing here is meant to represent the DNA. And then this thing here is meant to represent the polymerase. And this thing here is meant to represent DNA modification. So what we can study on the DNA level is, of course, mutations. So differences in the code here. But then something like chromosomal aberrations, of course, like do we have multiple ones? Did we lose something? Did we lose chunks and so on? And is our DNA modified in any way? So our polymerase makes DNA to mRNA. This is here, the thing is supposed to be mRNA. This here is supposed to represent splicing. So we can study all kinds of stuff there. We can study the expression level, different isoforms resulting from different splicing, and so on. Again, mutations, we can also do this on the RNA level. We don't have to go to the DNA level to study mutations, so differences from the normal reference genome. All kinds of editing that might happen here and so on. And then on the last level before we go to proteomics, we can do ribosome profiling, which is actually quite an interesting technique. So this thing here is meant to be a ribosome, and it sits like a bun around a hot dog here on this mRNA. And what you can do is that you basically fix this here so it cannot move anymore, and then you digest all the RNA. You get this tiny little chunk here in there left over, and then you can sequence this. So by this, you then know how many ribosomes were on your mRNA and where exactly they were and so on. So what you can do with this is that you can study, okay, how many ribosomes are on my mRNA? How does this differ between mRNAs and so on? Where on my mRNA are ribosomes most likely to be and so on. So you can do all kinds of interesting things with that. And in this talk, I mean, you can study all these things and much more, but in this talk, I'm going to mention 3 different aspects. So basically how we can include information about mutations on the DNA level into our proteomics search. And then some comparison of expression level on the RNA level, on the ribosome level, and on the proteomics level, because it actually is a difference and they correlate, but it's not the same. So you need to do proteomics because basically DNA doesn't tell you everything you want to know about processes in your cell. So let's start with just some mutations. So why would we be interested in mutations? Basically, because what we do in proteomics, and we've seen this throughout the last days, is we search against a database that is typically constructed of a reference genome, and we look for good matches for our peptides. But what happens if we have a mutation and we are not like the reference genome? So, I mean, we all are different humans and we all are not the reference genome. The reference genome is made to 70% of one individual, which is of African European background, male. So, I mean, most of us are female, so it's definitely, I mean, not me, but you. This clearly is a difference. So it might be nice to include this information in your peptide search, so you will actually find more hits. So this thing here is just meant to represent that there are changes or mutations that can actually change the amino acid sequence. And of course, many of them might be silent. So what we want to do is basically discover stuff that is changing the amino acid sequence of our proteins and include them into our peptide search. So we first do basically some normal NGS protocol and then include this in our proteomics project in our peptide search. So this is the motivation, and last year, the student who worked mostly on this, Pavel, together with his master student, Max, they published this nice paper on this that exactly tells you how to do it. So if you want to do something like this, read this paper. It tells you step by step by step in a very simple way what you should do. So what's the idea? The idea is basically that you have your normal MaxQuant search and data analysis, but before that, you have your variant extraction. So variant extraction basically means I want to find out which variants different from the reference genome I have in my organism. So you do this first with transcriptomics, genomics, whatever. So you can do it on a DNA level, RNA level, whatever you want. And then you do this, you use this as additional FASTA file in your peptide search. So we had this FASTA file yesterday that Hamid showed you when you run your MaxQuant search, that you need your FASTA file including all the proteins of your genome. So basically this results in an additional FASTA file that in addition to the reference genome also has information about the variants you have in your organism. So it makes the search a bit better. So how does this work? Basically, you first do that. For that, what do you need? So from your sequencing project, you need, of course, the reference genome and the genome annotation and your sequence data. So all this is not done by us. It's basically done by the sequencing software you use. And then you use this as input to MaxQuant. MaxQuant translates it to a nice FASTA file that you can just use then in your in your peptide search. And then you just go on with your normal proteomics experiment. You put in your raw data, you do the MaxQuant search, and you can do all kinds of data analysis. And these applications are just 2 applications that we thought might be interesting for this kind of project, but I guess you can come up with many, many more. Okay. So how does this work basically? So these files that I showed here, the BAM file, FASTA file, and so on, you just put them in MaxQuant here. So you click this fancy tools button here, and then this pop-up appears magically. And you click on variation extraction, and here you upload all the files you need. You press start, and it will output a FASTA file for you that you can then use in your peptide search. So that's basically it. And then you just do your normal MaxQuant search. Okay. So why might this be interesting, or what can we do with this? First, let's just think, okay, does this actually improve our peptide search? Do we actually find more? And I think on the first day, Tami said that, yes, it does, but not so much. So it improves it a bit, but is it really worth it? So this is the benchmarking we did. So we used data from, or they used data from this paper here, which is a really nice data set that you can use for this. So they used 4 different digestion enzymes, and then always counted how many variant peptides did I find. So this bar here tells you, okay, I found 191 peptides if I used only trypsin. And this is the unique peptides. So I did find them if I used only trypsin, but nothing of the other options. So if I did use, so these peptides here were not found if they used lysine or all these other kinds of enzymes. And we used also combinations, so all possible combinations of 2 or 3, and also all proteases. So what can you see in this plot? Basically, you can see, yes, it works. So our peptide search gets a bit better. So this here is just summed up over all of them. And we can see that trypsin, yes, it performs best. So if you only have one protease, use trypsin because yeah, you find most. But also it tells you, don't use one protease, use multiple ones, because then you find way more individual and unique peptides. So if you want a good coverage and if you want to find many peptides, use multiple proteases. And also this tells us, yes, it works. We find more peptides than without our variant extraction. And then what can we do with this? So for example, we can do something like this, and this also shows us, let's say, the challenges that we still have to meet when we do this kind of research. So this tells us, okay, from our transcriptomics, we know how often which mutation occurred. So this is like the reference amino acid, and then a mutation to the one we observe actually in our transcriptomic study. So here we changed from E to A and so on. And the color tells us how often, in how many cases we detected this actually in our proteomics experiment. And they are all blue, so they are around here. I think the median was something like 35% or something. So there is still a lot to improve. But also this tells you that there is a huge difference between transcriptomics and basically proteomics. So if you really want to know what is happening in the cell on the protein level, do proteomics and don't just rely on mutations or whatever. And the width just tells you how many we found. Okay, so this tells you that there's a lot to improve, but also that there's some really interesting processes going on. Okay. Back to that. So that was just to confirm, okay, it actually helps a bit, but I mean, more than that, beyond that, what can we actually do with this? So beyond just making our peptide search better, what can we do with this? And what we thought, for example, might be interesting would be to investigate, okay, if we have 2 different versions of a gene on our different alleles, can we count or can we look at how many copies of which do we have if we have more than 2 chromosomes? So what we looked at was basically now mutations and also the RNA level. So what we looked at was, or they looked at, was the HeLa cell line, because the HeLa cell line is basically known for having all kinds of chromosome duplications. So this is chromosome 8, and we have 3 of them. So now it would be interesting to see if there is a gene with different variants, which variant occurs more often. And this might, for example, be interesting for cancer research, because you might have this in cancer and you want to know, okay, which variant is there twice and which is there once, for example. So we have this mutation here where our reference says GAG, and our alternative says GAC, and we want to know now, okay, do we have a ratio of 1 to 2 or 2 to 1, or what is it? And this we can simply do on the RNA level or on the protein level, both. So what we can do is we just divide the number of reads we get for the reference by the number of reads we get for the alternative. We do the log2, so we get a nice number. And we can do that, or we can do it on the protein level, and we just sum up over the intensities, which is, more or less the equivalent of the number of reads, or like the most comparable value or score metric that you can use, and you sum up over fraction charts and modification. So you do exactly the same, and then you can plot them or their ratios in a 2D chart. So that's this, where we have this ratio for our proteins here on the y-axis and for the RNA on the x-axis, and they nicely correspond. So what you have is -1 for both of them. So what does that tell you? Basically that you have a ratio of 1 to 2, because we did the log2, and that's -1. So we have 2 times our alternative version and once only our reference version. And the RNA level corresponds nicely with the protein level. So that's nice. We can also do it with something that has more, like for example, here chromosome 11 has 4 versions. So there are all kinds of ratios you might see. So 3 to 1, 2 to 2, 1 to 3, or maybe they are all the same. So you can do the same here. And here basically we see that the ratio is 2 to 2 because it's here in the middle, our distribution. And I mean, it's a distribution because we did this for many experiments, of course. Otherwise it would just be 1 dot, obviously. But what you can also see is that the RNA has a smaller standard deviation than the protein. So there's still some improvement possible on the protein level. But also what might be interesting also as well is, so what if we have outliers on the protein level that do not correspond to the RNA level? That might actually tell you something about the biology going on there. That, for example, if you have something that where you know from the RNA level, all the variants are an alternative, but you don't see them in the protein level, maybe then it just doesn't fold correctly and you cannot observe it at all on the protein level. So what might actually be interesting is not the nice plots here, but the outliers that are not on this diagonal line, but that are something like here or here. So if we do this now for all chromosomes, we get this Mary distribution and we see, okay, nice, it is more or less a diagonal, but we can actually see that there are some outliers that are not corresponding between RNA and protein level. And that might be something interesting that one would, for example, want to study. So we think that might be something that is interesting or might be interesting for you. And of course, if you have compute number 3, there are 2 possibilities, 1 to 2. So this is this, or 2 to 1, which is basically here. So it makes sense. Okay, so I think I'm a bit fast, but that's okay. What else can we do? We can just ask the question, okay, if something is expressed a lot on the mRNA level, does all of this get to a protein or not? So if we use our expression level on the mRNA level as proxy for protein abundance, is that really a good idea? And I think we saw the answer also yesterday in Jürgen's talk, if I remember correctly, that it's not such a good idea. So let's see. So what do you want, if you want to do something like this, what do you do? So basically you have from your sequencing experiment, you have a FASTQ file, which is the raw file, which is basically a FASTA file. So it has a header and a sequence. And because it's a FASTQ file, it also has a quality score. So that's what this Q stands for. And then you align your raw file to a reference genome. You get the reference genome from the database of your choice, and then you align it. And so you know which genes you have in your sample. So this not yet we can do in Perseus or MaxQuant. We are working on this, but not yet. But what we can do then is basically, so the step from, if you already have an alignment of all your reads, you can put this into Perseus, and then Perseus will calculate you a gene expression table. So basically what does this do? So you have reads from your sequencing machine and you align them to the genome. So you have something like a distribution, how often you saw this gene in your cell, because you have many reads, so many chunks that you observed of this gene in the cell, or very few ones. So you know, okay, probably this gene there was transcribed a lot because I see many chunks of it, or many times the same chunk, or the other way around, I don't see almost no one, so probably it's not very highly expressed. And this you can do in Perseus, and then you get basically a table where for every gene you get a number that tells you how highly this is expressed. So you can just do this here in Perseus. You open this NGS data upload file. You insert here the BAM file. Here you just add it here. You press OK and it works. And then you also have this strand specificity option. So what's that? So basically maybe, so I mean, obviously you know that you can align what you read from your project, from your Illumina machine or whatever machine you have. You can align it in 2 directions to the genome, but maybe you're only interested in 1 direction. So maybe you only want the forward strand or the backward strand. But honestly, in most cases, it's going to be that. But you have this option to do it in Perseus if you want. And then at the time this was published, our labs, like the students at the time, compared it to something that was up to date then. So this was something doing exactly the same, developed by a different lab. And we basically see that the correlation is extremely high. So this is just to tell you, okay, we do the correct thing. You can do it in Perseus. You can also do it in other software, but it's the same. So why not do it in Perseus? And now this is what I was talking about, and this might actually be interesting. So what can you then do with this? So you can compare proteomics here with all kinds of other stuff. You can compare it to transcriptomics, so basically the RNA level. But you can also compare to the translatomics. So that was this ribosome profiling I was talking about. So basically, how many ribosomes are sitting on my mRNA of this gene at the moment. And what you see actually more or less makes sense, I think, that the correlation of proteomics to transcriptomics is there, but not more. But it's actually okay between translatomics and proteomics. So the translatomics tells you much more about protein abundance in the cell than just looking at the mRNA. So using mRNA and all kinds of RNA-seq and whatnot all as proxy for protein abundance might actually not be such a good idea. Yeah, and then here the highest R squared is, of course, between translatomics and transcriptomics. So there is a lot to discover in this whole process from DNA to RNA to proteomics. And for example, one study that I find quite nice, what they did is they were interested in exactly this. So what they did is that they knocked out a chaperone. So this thing here is a chaperone. And they were interested in, okay, I don't see any changes on the DNA level, but of course, if I knock out a chaperone, I will see changes in the protein level. So what they did is they compared for all kinds of genes of interest, the changes between the knockout and the control on the protein level and on the RNA level, basically. So here you have I think that was actually not RNA level, that was ribosome profiling, I think. So basically what you can see is here that doesn't change. So all the green dots here are things there where more or less the translation level doesn't change at all. It's around 0, so the ratio is more or less the same. But what you can see is that the protein abundance here changes a lot. So basically all these proteins you don't see in the proteome, but you can see that at least it's tried to translate them. So it makes sense when you study a chaperone, because these proteins then probably were not folded correctly, so are then disintegrated and degraded super quickly and are almost not found in the proteome. So this kind of study, I think, might be super interesting to do with this. Yeah, so we have seen 3 examples of what we can do with this, but I encourage you, do more. This is a super interesting field. So if you want, just try out all kinds of different aspects. It's really something that is understudied, in my opinion. And in the long run, of course, we don't only want to include NGS data, but we want more. We want also to include all kinds of other omics, so translatomics, proteomics, but maybe also something like metabolomics. I think we had questions about this already yesterday, if we're going to do that. So yes, we are working on this. And this year, we published, or Hamid, Daniela, Maria, and Jürgen published a nice paper with a plugin for, with this Metis plugin, basically, where you can include some, at least, metabolomics data, and we're also working on including the microbiome and so on. So that's our dream, that you can include all kinds of omics and cross-analyze this in Perseus and do more of these comparative studies that I just showed you some examples of. So with that, thanks to mostly Pavel and Max to develop this, most of the stuff I just showed you. So this is Pavel and this is Max, Jürgen, and of course, all of the other people in the lab. And now you can ask questions if you want. That's it.
Well, thank you for the talk. I have a question about the variants or mutations. Do you have to, I mean, you have very nice plots showing correlation between the,
what do you mean?
when you have chromosome 8 and chromosome 11 here.
Mm-hmm.
So how deep do you have to go at the protein level to find these variants?
Yeah, obviously the deeper the better, but I'm sure this is from fractionated samples.
Mm-hmm.
And I think you also need this because as you are very well aware, we're still lacking in sequence coverage, and sequence coverage is of course crucial if you want to catch all or as many as possible SNPs.
Yeah. Okay. So you would recommend fractionated?
Yeah, definitely.
I was wondering if you ever show that the protein hits that you get actually correspond to the variants within the FASTA file of like a particular mutated sample. If you have a protein hit and a variant hit, it's like a protein corresponding to an RNA that's supposedly mutated, do you actually ever demonstrate that the protein has the variant that is present within the alleles of, or within the variant of the sequencing data?
Do you mean with other means than mass spectrometry, like independent check, or what do you mean?
Well, so you have a protein and that will have some variant that you observe, and then do you actually know that that is present within the sequencing data? Do you like actually check that? You just kind of assume that it's true?
In the sequencing data of the particular person or cell line, or you mean in general in sequencing databases?
Yeah, either one. The one that you use within MaxQuant to do the matching.
I mean, so the way it works is that we have, in this case, we have exactly the same sequencing data of the HeLa. So these are HeLa cells, and we have the HeLa genome basically as the background from which we extract the coding SNPs with respect to the reference, like the plain human reference genome proteome.
So you don't extract variants from the protein level, you know them from your transcriptomic study, so you can include them in your peptide search.
Yeah, exactly. That's the second step. The first step is we extract the differences of the HeLa genome to the reference genome, and these are then put in a special way into a new FASTA file that we offer then for the proteomics analysis. And it's not, we don't incorporate these SNPs or any variants in the sequence, but it's actually written into the header of the FASTA file. And then we have a special proteomic search, which treats them almost like you would have modifications there. So it's basically also going to the combinatorics of differences between the reference and the HeLa genome. So basically they're put into the search, and the information comes from DNA or RNA sequencing. And in this case, it just happened to be the HeLa genome that we used there. But then in more relevant, like clinical studies, that would be the individual genome, however you did it from the patient, basically, that you use there. And so basically then these these coding SNPs end up in the search space for max quant for Andromeda. And then either you find them or not, basically. So they're just treated as modifications, essentially, in the search. And so basically you identified them both. You identified them on the nucleic acid sequence level, and you identified them on the proteomics level with our standards like 1% FDR and so on. And so the only thing that one might be thinking about is, so we don't have to be very strict in calling SNPs in the genomic data, because we anyway, so everything we find on the proteomics level is then confirmed with very stringent criteria. So what we usually do is that we set the thresholds for accepting, for calling SNPs very low. So basically just on a read count level, I don't know, 5 or whatever. So pretty lenient criteria for accepting the calls on the genomics level, and then very stringent confirmation on the proteomics level. So we have actually the data on both, right, for these.